Each year, there are between 250,000 and 500,000 new spinal injuries around the world. Currently, the only treatment applicable to these injuries can only reduce the deterioration of the neuronal networks linked to them, meaning it never manages to regenerate these networks, and it is also a treatment that can cause significant side effects. That is why this multidisciplinary team at Príncipe Felipe Research Centre, in collaboration with the Step by Step Foundation and Vall d'Hebron Hospital in Barcelona, has created a new treatment that combines cellular therapy with a nanoconjugate and obtains more effective results with a unique treatment.
The cells survive better, they get integrated into the tissue much better, and the compound that is released for a longer time allows the environment to be more permissive to the regeneration, to the axon regeneration, that is what actually need to really recreate again a new neuronal network.
With the design of this nanosystem, we are able to specifically trigger the release of the phosphodil intracellularly when the drug is reaching the lysosome, and in a sustained manner. So the cells are seeing phosphodil for longer time, so the bioavailability, as it's prolonged, the effect in the cell behavior, and afterwards the engraftment of the cells in the spinal cord lesion, it's much more efficient.
The main challenge lies in the complexity of each spinal injury. They are multifaceted injuries that cause tissue degeneration, a massive loss of cells, and damage to all neuronal cells at the same time. In order to recover function, the patented treatment uses a compound Fosudil, a conjugate with a polymer carrier through a self-immolative linker that breaks in the presence of a reduction medium or a high concentration of the reduced form of glutathione or GSH. Cell lysosomes contain high concentrations of GSH, so when the nanoconjugate compound enters the cell lysosome, the link breaks down and slowly releases Fosudil. Just a few minutes after introducing the nanoconjugate compound into the culture, the cell begins the process of endocytosis and processes it. The compound starts to be released and can be used by the cell that processed it. 24 hours after treating neural stem cells with a Fosudil conjugate in a culture, the cells are stimulated to mature, generating immature neurons and glial cells. That is when we apply cell therapy. Neural stem cell transplants treated with a nanoconjugate in advance survive better in the toxic environment of the injury, and the cells integrate more efficiently in the neural circuit, thereby improving its neuroprotection and regeneration activity.
Just with one administration, we are able to maintain the availability of the compound for a long time. This is not possible with the compound without the conjugation, with this nano formulation that we have established, because the compound is very unstable.
So both parts are ready. We have all the preclinical functionality done in acute and chronic spinal cord lesion preclinical models, and we are ready to have more funding to start clinical trials in collaboration with the Vall d'Hebron Hospital and Step by Step Foundation.
This is the first time that a patent has recognized the effectiveness of combining cell therapy and nanomedicines, and the results that it offers are promising. The nanoconjugate compound has proven to be effective in treating spinal injuries, but it is also having good results in other treatments. This nanoconjugate has proven to be useful both as a single agent and in combination with other drugs in order to inhibit the progression of metastasis in different tumors. In fact, there are already proofs of concept in breast cancer.